I'm excited to introduce our first speaker, Nima from Tahoe Therapeutics. Nima, please come up here, and we can get started.
It's very nice to be here. Thank you so much for the invite, Sam. I'm Nima Alidoust. I'm one of the co-founders of Tahoe Therapeutics. What we work on is basically mapping how various types of chemistry interacts with various types of biology. And by chemistry here, we mean drugs of different modalities, drug molecules that can become drugs of different modalities. And by biology, we mean different genetic backgrounds, basically patients that have very different profiles when it comes to their genetic and transcriptional backgrounds. But I'll say more about that as well. What we are very excited about is to understand heterogeneity in disease. And the way we like to talk about it is to say, okay, cancer is a polygenic disease. Let's actually treat it as such. And I'll explain a little more what I mean by all of those. Most of the common paradigms that are being used right now for treating cancer primarily, what they do is that they focus on a single isolated target. So basically, we oversimplified patients to a driver mutation, and then we take the corresponding protein, and we hope that if we bind some chemistry, if we find a chemistry that binds to that protein target, then we can shut down all of those erroneous signaling that's happening within the cell that leads to proliferation of cancer cells and therefore cure the disease. So in a sense, what we are doing is that we are taking a very monogenic view of cancer as a disease, but in reality, it's very far from reality. Most diseases, and cancer particularly, is a highly polygenic disease. What does that mean? It means that two tumors that are coming from two different patients, even if they're coming from the same organ, even if they're sharing the same driver mutation, they're not identical, and they won't respond to the same drug in a similar way. And we have seen this in many different trials when it comes to targeted therapies. The KRAS G12C drugs, for example, they were supposed to, for example, treat around 15% of patients with non-small cell lung cancer, but they end up actually treating only around 3 to 5%, depending on how you're counting. And the reason for that is that mutations co-occur. So it's not just one driver mutation that is driving the growth of the tumor. And this co-occurrence of genetic mutations, it leads to a basically combinatorially large genetic landscape that defines the molecular mechanisms by which cancer is actually evolving within the body. And what makes this even more complicated is that genes don't stay silent when another gene's activity is inhibited or perturbed. So basically, in response to the shutdown of a certain pathway, other pathways form compensatory mechanisms that would end up showing up, for example, as adaptive resistance. So this is a very complicated kind of environment where this is very complicated mechanisms that are happening within cancer that actually leads to an explosion of heterogeneity, even for patients that are seemingly very similar to each other. Now, one thing that has happened over the last decade or so is that we have actually gotten to understand a lot of these molecular mechanisms molecular signatures and molecular mechanisms a lot better. So right now, tests like Foundation 1 or Garden 360 are becoming table stake both as for diagnostic purposes and also for companion diagnostic purposes. So people are now empowered using some of these tests or other tests to understand what exactly their cancer looks like, what exactly their tumors look like, what genetic mutations have happened there, what are the broad specs associated with their cell states that they're dealing with. But this visibility hasn't really translated into what I guess in this conference we're calling genetic agency. What does that mean? It means that regardless of the mutations that they're seeing within their tumor samples, they can't really translate that into action. There are not therapies that they can take advantage of, therapies that are particularly designed for the specific broader subtype in which their cancer fits into, and therefore they're stuck with the same standard of care that's typically pretty poor performing as as everybody else. So in other words, we have not been able to translate visibility into these molecular signatures of cancer into actual agency when it comes to treating those various types of cancer. Now, the core belief that we have at Tahoe is that agency will come from our ability to measure better how perturbations actually interact with cells from various types of patients. So the challenge that we have in designing new therapeutics for these kinds of different subtypes that are usually subtypes that we don't even know about is that we actually don't even have a way of understanding how drugs interact with cells from very different genetic or transcriptional backgrounds, and therefore we don't have the ability to design therapeutics that are precise enough so that they actually have a high depth of efficacy in these subgroups of patients. And to make the matters even more difficult, it's the fact that if you are developing precision medicines, they can't be so precise that the patient population is so low that then it would make it very difficult to justify the economics to go to the trial and show that they actually work in those patients. So what we have been doing at Tahoe is that we actually have built a platform that allows us to test any drug or any chemistry in general on cells from hundreds of patients in one experiment. And that experiment could be in vitro or in vivo experiment. And that allows us to generate this very high content single cell transcriptomic data sets that measure how drugs actually work within these different genetic backgrounds. An example of one of the work that we have done is this data set that's called Tahoe 100, which we open sourced a few months ago. It's 100 million single cell data points. It's around 50 times larger than all other perturbative data sets that have been generated over the last 10 years combined. It is measuring how around 1,200 different treatments impact cells from 50 different genetic backgrounds. These are, when I say cancer models in this context, is cell lines, but these cell lines are coming from a variety of different indications, all major indications, colorectal, lung, etc. And that has allowed us to really have a very deep understanding of how different compounds that are in this data set interact with cells from these different backgrounds. Now, what I'm going to tell you is one story around some of the discoveries that you can make when it comes to precision medicine that are completely non-obvious and they could actually be life-changing for patients, and how we are using machine learning to actually extend a lot of these discoveries. So the name of our platform is Mosaic, and that's what you see up there. The Tahoe 100 data set that I just showed you in the last slide, it basically contains it basically produces a matrix. That's the effect of different compounds against different genetic backgrounds and different cancer models. In this specific case, what I'm showing you is a different data set is that we haven't open sourced. But what we discovered in this other data set was that a subset of colorectal cancer patients are responsive to a drug with a well-known mechanism of action in a way that was previously unknown. And when we actually used the transcriptional data that we have generated together with the metadata associated with those cell lines, and then we used our machine learning models to actually figure that out, what we found was that if the patients that are actually responsive to this drug with well-known mechanism of action, they have a mutation in one pathway but do not have a mutation in another completely orthogonal pathway. So the reason we can capture this is the access that we have to this heterogeneity of patients, that heterogeneity when it comes to genetic heterogeneity of the patients that we are experimenting with here. And what that led to was it led to a completely new understanding of how these different pathways are actually interacting with each other to drive the tumors. For most of the patients I'm talking about here, they were thought to be, their cancer was thought to be driven by KRAS. But when we actually, this new finding gave us a new understanding around how two canonical pathways that are seemingly unrelated to each other connect with each other to drive these different types of tumors. And the molecular data that we have also gave us the ability to identify what are the compounds that you have to mix with each other in order to kill these cancer cells. I won't go too far into details of this, but if you actually take one of the patients, one of the models that has the genotype that we had discovered, you put this combination into that patient, you see very strong tumor regression. And we have shown us as well that if they don't have that genotype, you don't see the regression. The standard of care for these patients is chemo. And in preclinical models, you actually see that the tumor continues to grow. It just is inhibited a little bit, but you don't see regression. And this is for 40% of colorectal cancer patients. 40% of cancer patients, they have actually that genotype that we've discovered. So we are generating these massive data sets. And what this is allowing us to is to identify precision medicines that are tailored towards the subset of patients that are within our data sets. But the reason that we actually have started to generate these kinds of data sets is because they actually can help us develop these models that we call them cell state models or single cell foundation models that allow us to generalize beyond the data that we are generating within our lab. And we have done that. We recently, it was a couple of weeks ago that we open sourced our Tahoe X1 model. It's a 3 billion parameter model. It's one of the most efficiently trainable models out there, which actually has allowed us, with very few experiments, to generalize to entirely new patient context. So the data sets that we have right now, we can predict how a certain drug molecule works in cancer models, basically, that are coming from different patients that have a completely different state than the patients that we have actually in our data sets. We are not stopping there. We are going beyond cell lines right now, and we are generating data sets in patient-derived organoids, in Vivo models. That's the core of our platform, actually. We were called Vivo, various types of phenotypes, and all of those are feeding into these models and allowing us to increase the translatability of the output of the models that we are generating as well. But because I've run out of time, I won't go too much into the details of that. Thank you so much for listening, and happy to answer any questions after the talk.